So welcome to this video on isoelectric point of amino acids and Zwitterion formation. In this video you will learn a relationship of the isoelectric point or Pi with pKa and pH, and also you will learn how pH affect the ionization of amino acids, leading to the formation of different dominant charged forms including Zwitterion. In addition, you will also learn how to calculate the Pi of given amino acids in a step-by-step manner. So what is isoelectric point? In order to understand isoelectric point, you should know the basic structure of amino acid and the groups, ionizable groups attached to the alpha carbon atom. In every amino acid, at least you have two ionizable group. The first one is alpha carboxylic group, the second is alpha amino group, and not in all, in about 6-7 amino acid you will have the side chain, ionizable side chain group as well. So that each ionizable group in this molecule has a characteristic pKa value. For example, for carboxylic group pKa is equal to 2, for amino group pKa2 is 10, and the value of pKa3 will vary depending on the nature of R. We will discuss that later. Now these pK values, the pH at their characteristic pK values, these molecules are ionized in such a way that their protonated and deprotonated forms are in equilibrium. It means the charge on the molecule is dependent on the pH, and at the pH, when the molecule become neutral, that pH is known as, that pH point is known as isoelectric point. So isoelectric point is the pH at which neutral form of amino acid is formed. It is a neutral form because the positive and negative charges are in equal amount. Such a species is known as zwitterion. So it is very important to understand the significance of isoelectric zwitterion in the calculations of isoelectric point. The other thing is you have to also understand the relevance of pKa with or to relevance to the in the calculation of isoelectric point. So you have seen that as you change the pH, the charge on the molecule or amino acid is changed. So the question is how the charge is affected when you change pH around its Pi. When you have seen that at pH equal to Pi, an amino acid has a net zero charge, or you can say a neutral molecule or zwitterion is formed. This is called as isoelectric pH or isoelectric point. When the pH is less than Pi, an amino acid has a net positive charge, and when the pH is more than Pi, an amino acid has a net negative charge. To further understanding isoelectric point or Pi, it is important that we identify pKa. I will explain pKa with the help of this famous equation of Henderson-Hasselbalch, which is commonly used to calculate the buffer pH, which tells us that pH is equal to pKa plus log A minus over HA molar concentration of these species shown here. Let us assume that the weak acid is 50% ionized, or you can say that the protonated and deprotonated forms are in equilibrium, then pH become equal to pKa. So what is the pKa? pKa is an ionization constant which is equal to the pH at which the protonated and deprotonated forms of a weak acid are in equilibrium, or you can say that the weak acid is 50% ionized. How the ionization is affected around the pKa by pH we will discuss in the next slide. So let us explore the relationship between the pKa and pH, or in other words how pH affect the ionization of amino acid around their pKa. So as I mentioned earlier, in most of the cases you have two ionizable group. The first one is carboxylic group with a characteristic pKa or ionization constant 1 equal to 2. The second one is amino group with a characteristic ionization constant K2 equal to 10. In about 6 or 7 which we are going to discuss, there is a third ionizable group, R group, with a characteristic pKa3, which will vary or which will depend on the nature of R group attached to it. In this case we will discuss the the amino acids which are listed here: aspartate, glutamate, histidine, arginine, lysine. So basically what we will do, we will explore how pH will affect the ionization of an amino acid, leading to the formation of the zwitterion. That will help us to understand the calculation of isoelectric point or pi. So let us further explore a little bit a relationship between the pKa and pH, or how pH affect ionization of an amino acid around their characteristic pKa value. I have explained example of diprotic amino acid with pKa1 equal to 2 and pK2 equal to 10, right? So what is the relation, how ionization will affect if we increase or decrease pH around their characteristic pKa values, right? So what is the relationship? The relationship is that if you increase, if you decrease, if you go pH below pKa value, there will be protonation of amino acid, and if you go above the pKa value, there will be deprotonation of an amino acid. So in this case, let's see what will happen if we decrease the pH less than 2. You will get this form produced, which is protonated. So there are two groups, one is NH3 plus, another is carboxylic group. They are capable of donating 2 proton, and therefore this type of amino acid are called as diprotic amino acid. Most of the amino acid are diprotic amino acids. So the charge on this species is +1. Now if we increase the pH above 2, what will happen? This group will be deprotonated. Why carboxylic group will be deprotonated? Because this is stronger acid comparative to NH3 plus. So if we increase the pH, this will be the species formed here. It has plus charge, it has negative charge in equal amount, therefore the net charge will be 0. So this is called as zwitterion. This is very important step in calculation of the pi, that you understand or identify formation of the zwitterion. So basically at pKa2, at this pH 2, these two forms will be in equilibrium. This is what it means. Now if we further increase the pH beyond 10, what will happen? You will deprotonate this NH3 plus group. So what will be formed here? You will form this species. What is the charge here? It is negative charge. So you have three forms are formed. One is positively charged, another, this is fully protonated, and the last one is, or third one is negatively charged. This is fully deprotonated, and this is the form which is known as zwitterion. It carries net zero charge because positive and negative charges are in equal amount. So this point where this is formed is known as isoelectric point, and this corresponds to certain pH. We will explore in the next slide. So what is the relationship between the pKa and pi, or in other words, how you will calculate, how will you use pKa values to calculate pi or isoelectric point? You have seen in the previous example that the Zwitterion was formed midway between the pKa1 and pKa2. The values of pKa1 and pK2 are given, which is equal to respectively 2 and 10. And because this is being formed in midway between pKa1 and pK2, therefore the pi is the average of the two pK values flanking the Zwitterion. Therefore you can calculate by averaging the two pK value. So the pi will be equal to pK1 plus pK2 divided by 2. So in this case 2 plus 10 divided by 2 is equal to 6. So this is very simple way of calculating the pi. You have to identify where the Zwitterion is being formed, and you should identify the flanking pK values. You average those two values, you will get pi. In most of the examination you will be given pK1 and pK2, and you have to calculate the pi, so you just average out. But if you have three pK values and you are asked to calculate pi, it will be little tricky. I will explain you in the next slide how to calculate the pi of glycine. So glycine is a diprotic amino acid. This is the structure of amino acid. R group is H. You have pKa corresponding to carboxyl group is equal to 2, pK2 corresponding to NH2 is 10. So how you will calculate the pi is very simple. So the pKa1 is 2, pK2 is 10. So I have indicated here these values. So what will happen if we decrease the pH beyond 2? You will form this protonated form. The charge is +1. It is fully protonated diprotic acid. If you increase the pH beyond 2, this will deprotonate carboxylic group. This will be the species. This has positive charge, it has negative charge. The ionization will not be affected here, right? NH3 plus will remain intact here. Now if you go beyond 10, then NH3 will deprotonate further. Now in this molecule, in this form, the charge is 0. So this is Zwitterion. It has positive charge, negative charge in equal amount, which cancels out. So net charge is 0. This is Zwitterion. Here this is negative charge it carries. So this is fully deprotonated. This is fully protonated. So what will happen here? You have formed the Zwitterion. So the point corresponding to this is the isoelectric point or pi. How you will calculate? You will average out these two values. So pK1 plus pK2 divided by 2 is equal to 2 plus 2 here plus 10 here equal to divided by 2 is equal to 6. So it is very simple. You identify where Zwitterion is appearing and what are the flanking pKa value. You average these two and you will get the answer. Very straight. So now coming to the calculation of pi of basic amino acids. So in basic amino acid you have R group, which is amino group, basic group, right? So I have shown here RNH3. This means this is good for showing lysine and arginine both. So in this case you have 3 pK1 correspond to carboxylic group, which is 2. Amino group has pK2 corresponding value is 9, and the R group is basic and corresponding pK value is between 10 and 12. You can average out it to be 11. So remember these three pK values. pK1 is 2, pK2 is 9, pK3 is between 10 and 12, or you can average 11, sorry. So using this So this is 3 pK value. You will form 4 charged species, including one which is Zwitterion. So this is an example of multiprotic basic amino acid, how to calculate. So there are 3 pK values. pK1 is 2, pK2 is 9, pK3, as I said, I averaged 10 and 12, which is equal to 11. It's very simple here. So if you decrease the pH below 2, you will protonate amino acid. So this is fully protonated. This has 3 groups which can donate proton. This is called multiprotic. Now if you increase the pH beyond 2, this group will deprotonate first. So you have the the charge on this molecule is +2. If you increase the pH beyond 2, you will deprotonate carboxylic group, forming this species. What is the charge on this? You have +1. You further increase the pH beyond 9, you will deprotonate this group, not this one, right? So pK2, pH above pK2 will deprotonate amino group, and you will make this species which has +1, -1. So total charge is 0 here. This is the Zwitterion. If you further increase the pH beyond 11, you will get fully deprotonated carrying -1 charge. So you have 4 species. The leftmost is fully protonated, the rightmost is fully deprotonated, and the the Zwitterion is being formed in between the pK2 and pK3. So what is the pi? You can calculate by averaging the value of pK2 and pK3, which is 9 plus 11 divided by 2 is equal to 10. So let us calculate the pi of an acidic amino acid such as aspartate and glutamate. They are also multiprotic amino acid. This is the general structure of an acidic amino acid, aspartate or glutamate. This has alpha carboxylic group, pK is equal to 2, same. This has the side chain carboxylic group, pK2 will be 3, between 3 and 4, and alpha amino group pK3 is 9.6. So remember these 3 values, pK1 2, pK2 is between 3 and 4. You can use 3 or 4 or average, doesn't matter as long as you understand the method of producing Zwitterion. And alpha amino group has pK3 close to 10 or 9.6, right? So let us calculate the pi of acidic amino acid. So let us calculate the pi of acidic amino acid. There are 3 pK values. pK1 is 2, 3.6, pK2, and I have averaged out, right? And pK3 is 9.6 or 10, same thing. So basically, if you decrease the pH below pK value, then it will be protonated. So you have this is fully protonated. How much is the charge? You have +1 charge here. If you increase the pH beyond 2, this will deprotonate, and carboxylic group, this group will be protonated first, not this one. So you will make this species, and this has +1 charge here, minus here, total charge is 0. This is the Zwitterion, and this is what we are looking for. So if you further increase pH beyond pK2, you will deprotonate this group now, because this is acidic pH. So you have this species is formed. What is the charge here? -1. You still have NH3 plus group. So if you increase the pH beyond 10, you will deprotonate this group, and you will make this species here. What is the charge here? -2. So basically there are three pK value. In this case also you will have four different charge species, including one which is Zwitterion. So this leftmost is the fully protonated, the rightmost is fully deprotonated, and Zwitterion is being formed between the pKa1 and pKa2. So how you will calculate? You average the value of pKa1 and pKa2, you will get the answer. So Pi will be pKa1 plus pKa2 here. So what is the answer? 2 plus 3.6 divided by 2 is equal to 2.8. So this is very simple. So thank you for listening to this video. I hope this will help you, and if you think so, please like it, subscribe it, and share it with your friends. I'm sure you will find questions in your exam related to isoelectric point. Until next time, take care and bye bye.